was doing some of these biochem passages by myself and kind of talking through them out loud, so I might as well just do some of them on camera. So I'm going to scroll through the passage and give you guys time to answer the questions on your own. Okay, so I'm just going to go through it and kind of highlight the things that I would normally highlight and point out. So it says stomach ulcers have long been thought to result from an overproduction of HCl by the gastric mucosa cells. When drugs that neutralize or reduce the production of stomach acid are used to treat patients with ulcers, 95% of the patients had a recurrence of the ulcer within 2 years. So I like to highlight statistics because a question could kind of give me an idea about this, like which of the following could work or which of the following treatments wouldn't work. So here we see that drugs that are reducing the production of stomach acid, they are likely to have a 95% of recurrence of stomach ulcers in the patients. So it's not super effective. Scientists suspected that another factor was the true cause of ulcers. Helicobacter pylori was isolated from biopsies of ulcers. The organisms were found in greater than 95% of people with ulcers. When ulcer patients are treated with a combination of antibiotics and drugs, there's no recurrence. So we see that this is a better option. Originally, 95% of patients, they have a recurrence, only treating stomach acid. And then now we see that antibiotics and stomach acid reduction, there's no recurrence in the ulcers. The bacterium produces a highly efficient urease localized to the outer portion of the cell wall that converts urea into ammonia and carbon dioxide. And then it goes into some tests for H. pylori, and then it's talking about giving individuals radioactive urea, testing for exhalation, blah, blah, blah. And then this, I'm assuming, is talking about this figure. So 2 strains of H. pylori, 2 hours, subcultures, and then they're looking at the antibiotic streptomycin. I'm not going to read the table or the figure until I actually have a question about it. And then I'm just going to read the figure caption. So this is growth of strains A and B. So A and B are both strains of H. pylori in the absence and presence of streptomycin, which we know is an antibiotic. And then the arrow down here indicates additional streptomycin addition. So first question, based on the passage, which of the following methods would prevent the recurrence of stomach ulcer? Use of drugs that prevent the production of acid. I mean, we kind of mentioned this before, right? Because up here we see that neutralization or reduction of the production of stomach acid, 95% of the patients, the ulcers come back. So we're automatically going to remove that. That's not going to be the best case scenario. Use of drugs that neutralize stomach acid. Well, we don't want to neutralize the stomach acid. We just want to minimize its production, because we need stomach acid. Use of drugs that inhibit bacterial protein synthesis. Well, here, right off the bat, we can see that the answer we're looking for is that you want a combination of antibiotics, so bacterial protein synthesis, inhibition, and drugs that reduce acid production. And the best answer seems to be D. Okay, it is now generally accepted that H. pylori can cause ulcers. Proof of this most likely depended on the demonstration that. So to reword this question and really digest it in a way, I could just read the question and say, okay, H. pylori causes ulcers. Which of the following shows that? Which of the following answers would prove that H. pylori causes ulcers? So let's look at A. People with stomach ulcers have antibodies to H. pylori. This doesn't make any sense, because if you have antibodies to H. pylori, you would probably not have stomach ulcers. And this is kind of telling you the opposite. This is telling you that with the antibodies to H. pylori, so with resistance to H. pylori, you still have stomach ulcers? No, won't buy it. Healthy individuals have antibodies. I mean, sure, but I'm not 100% with this one, because just because a healthy individual has antibodies to H. pylori doesn't mean, this isn't telling me that H. pylori itself causes ulcers. So I'm going to cross that off. Ulcers could be produced in healthy organisms by infecting them with H. pylori. This one makes a little bit more sense. And then this is just talking about genetics. The organism can be passed from mother to fetus during pregnancy. So it's saying that bacteria will pass down, but that's not talking about ulcers at all, and we don't care about that. Dumb. Which of the following explanations best accounts for the results of Figure 1? Boom. Figure 1, scroll down. Now let's read what we didn't read before. So a scientist grew 2 strains of H. pylori A and B in a lab. 2 hours after the initiation of the cultures, each culture was split into 2 subcultures. One subculture of each strain was exposed to streptomycin. So let's zoom in here and let's read it on our own without looking at the answers or any more of the questions, so we have our own idea of what's to come. So here we see that the y-axis is the amount of cells per milliliter. So it's a quantity over time. So this seems to be some kind of rate, right? And now let's see the symbols. So we have strain A, which is this one down here, and strain A with streptomycin. They're both starting off at the same point. And then, like it says up here, they're split into 2 subcultures. So strain A, the normal one, just started increasing at a greater rate, the amount of cells present of H. pylori. And then strain A with streptomycin kind of was maintained the same. Remember that streptomycin is an antibiotic. So if it is resisting growth of H. pylori, that means that the antibiotic is doing its job, because that's what it's supposed to do, prevent the cell proliferation. So that means strain A has streptomycin working down here, and then the other strain subculture was increasing. And then we have strain B, which was just kind of exponentially almost increasing over time, and there's no separation into the 2 subcultures. So you see that even with the addition of streptomycin, the amount of H. pylori cells per milliliter were kind of maintained the same, and they were growing anyway. Okay, so strain A grows faster than strain B. Like we said, this is over time, so that could be the case because strain A has a sharper slope or a steeper slope. So that could be true. But let's find if there's maybe a better answer. Strain A grows faster than strain B. Okay, we already said that. And it's resistant to streptomycin. So strain A, if it were to be resistant to streptomycin, streptomycin addition would not cause the cells to decrease or to maintain the same. So antibiotic resistance basically means that the antibiotic isn't working and that there's still going to be an increase in the amount of H. pylori cells. And that's not the case, because here we see that strain A, it doesn't increase with streptomycin. It stays the same, which means that strain A is not resistant to streptomycin. So we can cross off B. C, strain B grows faster than strain A, and strain B is resistant. I don't think strain B is faster than strain A. Strain B has a narrower slope. It's not as steep of a slope as strain A. And you can actually compare values here if you wanted to as well, but I think that would take too much time. Strain B grows slower, so that's kind of telling me the same thing as A, and strain B is resistant to streptomycin. This makes sense because streptomycin, even when added to strain B, the cell count of H. pylori is still increasing, so it means streptomycin isn't working. Boom! I'm eating this shit up. Most bacterial cells and human cells are alike in. So for this one, you don't even need the passage information for this. It's literally asking you for the difference between prokaryotic and eukaryotic cells. So let's start with A, the ability to produce ATP via ATP synthase. I mean, all cells need ATP. They need energy, so this seems about right. The chemical composition of their ribosomes. I mean, we know that their ribosomal subunits have different sizes, so I'm going to assume no. Their enclosure in cell walls? No, not both of these have cell walls. The shape of their self-replicating structures that carry their DNA? No. Like chromosomes? No. I don't like that. Also, think about even like organelles. I don't believe it. Yeah. Which of the following experiments would best test the hypothesis that urease is necessary for the colonization of the stomach by H. pylori? Easy. This shit is easy. Which of the following experiments would best test the hypothesis? So, fuck the question. Forget about what the question is asking you. let's come up with our own question based on this. So we want to show urease is necessary for H. pylori to fuck shit up. So which of the following answers is showing that urease is necessary for H. pylori to colonize your stomach, right? So let's zoom in here a little bit. Exposing ulcer patients to antibodies of urease. So ulcer patients theoretically have H. pylori. If you're exposing them to antibodies of urease, I mean, I guess that would like help cure it, but that's not telling me that urease is necessary. I don't know. It kind of sucks. I'll leave it there. B, exposing uninfected animals to a strain of H. pylori that lacks urease. So if you are exposing animals with no urease, with H. pylori and no urease, and they still have colonization of H. pylori, I mean, that would show that urease is necessary. If you expose uninfected animals to a strain of H. pylori with no urease and the stomach is not colonized, that would show that urease is necessary. Exposing ulcer patients to radioactive urea. I mean, I remember reading about that over here. Yeah, radioactive urea, that's a test for H. pylori. That's not talking about urease. Boom, removed. Don't care about you. Measuring urease activity. That doesn't mean anything to me. Just because there's urease, that doesn't mean it's necessary for H. pylori to colonize the stomach. So C and D don't make any sense. Now, A, if you have an ulcer patient and they have H. pylori and it's invading their stomach, if you give them antibodies for urease, that's not telling me that urease is necessary. But if you expose animals that have H. pylori, if you expose the animals to H. pylori and no urease, you can see that the H. pylori is probably not going to be as effective as at colonizing the stomach. So I think that exposing uninfected animals makes more sense on a test scale because A is not necessarily showing that urease is a necessity. I wanted to do that passage and just kind of break it down because I love watching the AAMC breakdown videos and talk them through and see why I thought the way that I did, and maybe that's helpful for you guys. But if you guys want to see any similar videos or more AAMC breakdowns, yeah, let me know because it helps me to be able to answer the questions and explain and cross my fingers and hope that I'm not getting it wrong as I go on. But I hope that this breakdown was useful to some extent, and yeah.